Hey everybody, welcome back. Today we are going to look at Unit 6, Lesson 1, Exploration 2, titled The Structure of DNA. In Exploration 1, we looked at a few of the scientists that were key to discovering that DNA is in fact the macromolecule that contains an organism's genetic information. And today we're going to talk about what that structure of a DNA molecule is and a few of the scientists that played key roles in discovering what that structure was. If you remember from last time, Hershey and Chase were two of the scientists that played key roles, and after their experiment, the scientific community accepted that DNA was in fact responsible for the genetic material, and so now the race became to figure out what it actually looks like. All right. If you think back to an earlier unit, I think from Unit 2, where we looked at macromolecules, deoxyribonucleic acid is a macromolecule that is made up of structures called nucleotides. If you remember, a nucleotide has three parts: a sugar, a phosphate, and then a nitrogen base. And those three components are what make up that nucleotide group. Now, in DNA, there are four different types of nitrogen bases. Every nucleotide in DNA has a deoxyribose sugar. Every nucleotide in DNA has a phosphate group, but the nitrogen base is what's different, and it's what gives DNA that coding ability. It's what gives it the language that allows it to give all of the instructions for how to make all of the proteins in your body. Now, we call those four bases, their names are cytosine, thymine, adenine, and guanine. Now, we'll talk a little bit more later about how those fit together, but for now, those are the four. And we are going to shorten those down to C, T, A, and G. Now, it's important to note that there are two basic structures. These four nitrogen bases have two basic structures. This picture does a pretty good job of showing it's either going to be a single ring structure, like you see here. These single ring structures are what we call pyrimidines, or the nitrogen base is going to have a double ring structure. You kind of see a ring there and a ring there, same thing here. Those are called purines. Now, the reason why this is important is that these nitrogen bases are going to fit together kind of like puzzle pieces. Another way to think about it would be like the hooks on a zipper. They're going to link together because their shapes are complementary. If you look here, A and T have a complementary shape. C and G have a complementary shape. And again, we'll talk about that more right now, actually. Another one of the scientists that played a key role in this process of discovering DNA was a man named Erwin Chargaff. And Erwin Chargaff set out to try and figure out if the nitrogen bases in DNA were random or if they were intentional. And so he did an experiment where he looked at several different types of DNA strands, several strands of DNA from all kinds of different organisms. And you can see these six organisms right here. In every single one of these organisms, he noticed a trend. He noticed that no matter how many total nitrogen bases there was always the same, or at least a very similar ratio of adenine to thymine, in other words, A to T, and G to C. You can see if you look at these other types of ratios, they're not similar, but when you compare the ratio of A to T, they were almost always the same. G to C, almost always the same. And it goes back to that original structure. The purine and the pyrimidine have a complementary base pair. They go together, they fit together. And this was key because for a long time, people just assumed that there were random letters in the DNA code, but Chargaff was able to prove, or at least demonstrate, that there is some sort of rhyme and reason to how these fit together. Now, that was going to be very important to someone later on that we'll talk about. The next scientist that played a key role in discovering the structure of a DNA molecule was a scientist named Rosalind Franklin. And Rosalind Franklin was an expert in what's called X-ray crystallography. She was able to essentially take pictures using X-ray technology of extremely small objects. DNA is an extremely small object. And so she was able to generate a picture using an X-ray machine, and it showed enough detail that the pattern and the angle of that DNA strand could be seen. She was the scientist who was later on credited with discovering the double helix shape of a DNA strand. Here's one of her pictures that she's famous for. This picture showed the scientists that were working on this problem enough detail to determine the angle of the twist, the space between the nucleotides, and the fact that it is in fact a double helix shape. Now, Rosalind Franklin's research was unfortunately not very well appreciated, in large part due to the fact that she was a woman. At the time, female scientists were not given the same level of respect as their male counterparts, and in fact, her research was actually stolen from her, and she was not given credit for it until after she had passed away. Due to her exposure to X-ray radiation, she developed cancer and died. And her research, now is found to be very important to the discovery of the shape of a DNA molecule, but at the time, not very many people gave her credit for it. Most of the credit for discovering the shape and structure of a DNA molecule is given to two scientists named James Watson and Francis Crick. Watson and Crick were given the task of developing a model, and they were eventually able to build their model, but they were having all kinds of trouble until they learned of the research of Erwin Chargaff and of the picture of Rosalind Franklin. It was only with these two additional pieces of information that they were able to create their model. Now, they were given all kinds of awards, they were given all kinds of prizes, and it wasn't until much later that they actually started to credit people like Rosalind Franklin for helping them in this process, because without her research, it would have taken much longer to develop their model, and they might not have been able to come up with it, at all. So, but James Watson, Francis Crick, those are the scientists that are credited with discovering the shape of a DNA molecule. So, just to recap real quick, Erwin Chargaff, very important contribution. He developed Chargaff's rules, which are the rules that say that adenine and thymine are complementary to each other. Guanine and cytosine are complementary to each other. Rosalind Franklin, X-ray crystallographer, took a picture of DNA that showed the shape and angles of all the twisting, and that picture was used by Watson and Crick ultimately to build their model of DNA to be able to show the world what this genetic material actually looked like. Okay, so let's talk about what it actually looks like. Now, I highlighted this whole paragraph because it's very important. The current model that we have represents nucleotides in a single strand that are joined together by covalent bonds. So one nucleotide to the next on that strand is connected by a covalent bond. And if you remember back to, Unit 2, covalent bonds are pretty strong bonds. They're where electrons are shared, not very easy to break. So the bonds that exist between these two nucleotides, between that phosphate and that sugar, phosphate and sugar, those bonds are very strong, not easy to break that way, okay? But each strand of DNA is connected to the complementary strand by something called a hydrogen bond. Now, if you remember back, hydrogen bonds are not actual bonds where an electron is shared. Hydrogen bonds are created by an electromagnetic attraction between polar molecules. So there's not actually a real bond that's formed here. It's more just like a magnetic attraction. It's very easy to form, but it's also very easy to pull apart. And that's going to be important as we talk about how the DNA molecule is actually used. But again, just make sure you understand this picture, okay? Each side of the strand is made up of nucleotides that are connected to each other by covalent bonds. The two strands together are connected by hydrogen bonds, much weaker, much easier to break, also much easier to reform. Now, The side of a DNA molecule is called a backbone. It actually kind of even looks like a vertebrae. So we call this the backbone right here. All right. And then again, if you look at this picture, it's going to show you those nitrogen bases, A and T, C and G, they are always going to be complementary to each other. When you have a G on one side, you'll have a C on the other. When you have an A on one side, you'll have a T on the other. And that's going to be, again, very important as we begin to talk about some of the other functions of a DNA molecule. All right, so that's it for this lesson. Make sure you go back and understand the scientists that are involved, and specifically pay attention to this paragraph because it contains key information about the shape of that DNA molecule.